Welcome science people to the iTrial Biotechnology channel. My name is Ali Fukutoabe, and in this video we're going to talk about a final review paper project that me and my group of authors made, with the title of Analysis on Current Development of Bioproduction Strategies for Propionic Acid. Without further ado, let us start. Well, if you haven't heard about propionic acid is a distinct short fatty acid known to occur naturally in the environment. Another fact is that propionic acid is categorized as carboxylic acid due to the carboxyl group. The application of propionic acid has been utilized in the production of various types of products throughout the industry fields, such as usage in pharmaceutical drugs, organic or natural-based herbicides, use for flavor additives, preservatives, and antimicrobial agents in the food industry, and lastly involved in the process of thermoplastic productions. And there are still many more applications of propionic acid that haven't been mentioned here. Oh, and one more thing, the usage of propionic acid has been approved by the FDA. The high demand of propionic acid from the worldwide market has resulted in an estimation of 470 kilotons in 2020. The demand has been significantly increasing throughout the years before. So propionic acid has been commonly produced using petrochemical processes. However, this has grown concerns due to the fluctuating price of petroleum and the negative impact towards the environment. Since then, it is considered that the production of propionic acid needed to shift into using more sustainable methods and utilizing renewable biomass. That is when biotechnology applications such as fermentation offer a gateway to a more sustainable production on an industrial scale. And that is when we call it bioproduction. Bioproduction or bioprocessing consisted of two main parts, which are the upstream processing, where cultivation of microorganisms and fermentation occurs, and downstream processing, where the propionic acid will be separated from the fermentation, purified, and finalized. During the upstream process, there are three aspects that needed to be paid attention that will affect the fermentation process of propionic acid. First is the microbial strain, second are the parameters, and lastly are the fermentation systems and bioreactors. These aspects will decide the maximum yield, titer, and productivity of the fermentation. Most common microbial strains that are used in this current times of study belongs to the Propionibacterium, such as Acidipropionici species and Freudenreichi species. Most of the Propionibacterium are known to use the succinate pathway for the extracellular production of propionic acid. For the parameters, there are 5 important parameters to be considered: temperature, pH, time of fermentation, carbon source, and additional nitrogen source. Attempts on utilizing renewable carbon source for fermentation have been studied, such as whey lactose, glucose, glycerol, xylose, corn cob and stover, artichoke hydrolysate, and sugarcane molasses. After we sort out the strains and the parameters, we then move on into the fermentation systems that will be used for the fermentation. These systems will consist of batch, fat batch, and continuous fermentations. Different bioreactors have also been used for the fermentation process of the propionic acid, from using standard suspension to immobilizations of the cells. When the fermentation is done, it will then undergo downstream processing that consists of 3 main processes: clarification, extraction, and ended with an optional further purification process. Clarification is done to remove big-sized particles such as cells from the broth. The 2 known methods for clarification are centrifugation and filtration. Then, extraction is done to separate the propionic acid from the fermentation broth or the aqueous phase. The known methods compatible for propionic acid are adsorption, electrodialysis, reactive extraction, precipitation, and membrane-based extraction. Lastly, further purification processes for concentrating the propionic acid can be done by removing most of the remaining byproducts such as water. Oftenly, distillation and evaporation can be used for purification in petrochemical-based propionic acid production. However, there is still lack of information for bio-based propionic acid. Well, it has been fun explaining to you guys about our paper. It is taken a note that this is still in development and further research is required to fulfill the sustainable production of bio-based propionic acid. Thank you guys for listening, keep studying, and be curious about science. Goodbye.